On today's Arabidopsis Research Roundup, we are discussing a paper which has just come out in PNAS, and it's a collaborative piece of work between the lab of David Balcom in Cambridge University and also the lab of Joe Ecker, who's in the Salk Institute in La Jolla in Southern California. And we are talking today to the lead author on this paper, Matt Lewsey, who is a British-born postdoc who's now working out with Joe Ecker. So the title of the paper is Mobile Small RNAs Regulate Genome-wide DNA methylation, and it's really a continuation of the story, I think, of how small RNAs are able to influence gene expression. So thanks very much, Matt, for joining us, and give us an explanation of this paper, please. Yeah, that'd be great.
Yeah, thank you very much. So the key thing we found here basically was that the shoot root mobile small RNAs are able to target DNA methylation genome-wide. Charles and Attila, who were authors on this paper, previously published a really, what I thought was quite an elegant study some years ago, showing that small RNAs will move from shoots of Arabidopsis down into the roots. And they showed that the predominant species was the 24 nucleotide small RNAs, which are known to be associated with DNA methylation. They were able to show at the time that a small number of transposons had DNA methylation regulated by these mobile small RNAs. But the limitation of the approaches available at the time meant that they couldn't look genome-wide. And so the kind of step forward we made here was to actually look genome-wide and understand the effects. And so this was a collaboration that, as you said, Joe and David established. And I worked closely with the co-lead author on this paper, Tom Hardcastle, who's an excellent bioinformatician and statistician in David's lab. Basically, we used the same nice grafting approach that Charles and Attila used. So they set up more grafts in the same way as in their small RNA paper, which allow us to logically deduce the movement of small RNAs and also the effect on DNA methylation. So if you take a wild-type root and you sequence out small RNAs, sequence out the DNA methylation in those, you'll have certain patterns genome-wide. If you then we use the DCL234 mutant, which is deficient in small RNAs. So if you then sequence out the roots of a DCL234 mutant, you've removed the small RNAs, you'll be able to see where they've gone from, and you'll be able to also see where the DNA methylation has been lost. Now, the neat part here is when you graft back on a wild-type shoot, you restore back the small RNAs, and you can sequence those back out in the roots, see which ones come back, and then sequence out the methylation, and you can see which patches of methylation come back as well. So now you know which ones are controlled directly by the small RNAs. You can also look to find patches of methylation that are indirectly controlled. So when these reappear when you graft on the wild-type shoots, but you can't find small RNAs associated with them. So you've got two different key types, the directly small RNA regulated and the indirectly small RNA regulated. So we set up these experiments, and the neat trick, the novel part here was that Tom established some nice statistical methods to enable us to look at the patterns of association between small RNAs and DNA methylation, scan it genome-wide and find which ones associated with one another, which ones were indirectly affected by small RNAs, which ones were directly affected. So when we do this, what we find is that there are thousands of loci that are regulated in this fashion. There are more in the indirect group than in the direct group, but you've got a lot in both camps. Okay. And these tend to fall into their predominantly non-CG methylation, so CHG and CHH context methylation, the kind of methylation that is associated with RNA-directed DNA methylation. So this is quite logical. We then went on to do some analyses on some mutants and existing data from the Jacobson lab, looking at patterns of DNA methylation in many, many different RNA silencing mutants. Yep. And we were able to associate our regions of what we'll call mobile methylation, the regions we'd found that were small RNA controlled, with loss of methylation in certain RNA silencing mutants. These mutants were known components of a particular branch of RNA-directed DNA methylation, DRM1, DRM2. So this allows us to pin down the regulation of mobile methylation to that particular branch of RNA silencing. Okay. And we also noticed, as we looked at the features our mobile methylation is associated with, we found that these patches are particularly associated with certain types of transposable elements. They tend to be small transposable elements, and they tend to be gene proximal.
Okay.
So they tend to be retro elements, mostly. Okay. Some small DNA elements as well. So this is interesting because these types of elements are the ones you would worry about if they became reactivated. There's a reason you might imagine to try and clamp down on their behavior, because if they're gene proximal and they become reactivated, you've got a problem. You can get genome disruption.
Yeah.
But the surprising thing that we noticed when we did this, we actually did sequence out transcriptomes from the roots as well, looking across the graft combinations. Mm-hmm. And the effects on gene expression were really quite limited. It was only a handful of genes that appeared to be regulated in any kind of mobile fashion. And so that leads us into, I think, our future work. Our expectation or our prediction is that we don't see strong effects on gene expression because we're not really looking at the right plant to do this in. Arabidopsis has a very compact, tightly regulated genome. It's relatively transposon poor, and the transposons are clamped down. They're not very active. Yeah. So in the future, we want to look at other species, for example, tomatoes, things like this, that have bigger, more transposon-rich genomes, where we anticipate we might see a stronger effect on gene expression because you've got more transposons and you might have more problems if they become reactivated. So this is where we intend to go from here.
Okay, so that's a great summing up of the paper, certainly very comprehensive. So just to get it clear for me and maybe for other listeners, so if you looked at the small RNAs from the root anyway, so there are species which come down from the shoot which aren't present in the root all the time, and those are the indirect ones. Is that the right way of thinking about it?
The indirect effects could be one of two things. So we see changes in DNA methylation that correlate with the loss of small RNAs, but no small RNAs specifically targeting that region of the genome.
Okay.
So what you might have there is some kind of intermediate messenger. So there's some direct effect of a small RNA at some other locus in the genome that then signals to this second region.
Okay.
Or it might be that we don't fully understand how small RNAs target in the genome, and so there are small RNAs that we've missed. Okay. I guess a third explanation, actually, now I consider it, is that you might have things below the threshold of detection of the sequencing technique. Yeah. So there were small RNAs there, but we couldn't actually get hold of them.
Right.
But yeah, basically what this comes down to is you've got methylation in the root, which changes when you remove small RNAs from the shoot, but those small RNAs do not target directly to that location.
Yeah, I see. Okay. And so one bit of work that just towards the end of the paper, which you haven't mentioned here, is that you use two different ecotypes. So you use the C24 ecotype and a Columbia ecotype, the roots and the shoots. So I think just to sum up, if I could, you showed that the C24 generated small RNAs are able to target normally non-methylated regions in the Columbia genome, if that's right. Yeah. So do you want to just touch on that a little bit? What was surprising in that work that you found?
Yeah, so this we thought was really nice, actually. What we found was you've got slightly different patterns of DNA methylation across Arabidopsis accessions, and in the course of experiments we grafted together C24 and Columbia, which have slightly different patterns. Mm-hmm. These differences get termed epialleles, so epigenetic alleles, different patterns of DNA methylation. Mm-hmm. We found that small RNAs specific to C24 can be transmitted from a C24 shoot down into a Columbia root, and they can target the C24-like pattern of DNA methylation at a small number of loci. So you can transmit these epialleles. You can take an area that's previously not methylated in the Columbia accession, and you can methylate it using the C24 accessions and make that small patch of methylation look like it's C24.
Okay. And is that a sequence-specific effect? So are there sequences that the small RNAs are binding to, or is this kind of this indirect effect which is occurring?
So these were direct. So we actually mapped the small RNAs to the location, and that's where the DNA methylation appears.
Okay. So in the Columbia background, there basically isn't the small RNAs either aren't there, or they're just not methylating? Like, maybe you don't know that.
So the small RNAs were absent in the Columbia background at the particular examples we found.
Yeah, okay. It's interesting, even between ecotypes, there's obviously such differences there. Continues the complexity with this small RNA silencing work. There's plenty more to be done along the way, I'm sure. Some good high-profile papers to come out yet, I think. So,
yeah, hopefully so. Certainly enough to keep us busy anyway.
Yes, indeed. Excellent. Okay, that's great. So thank you very much, Matt. So I've just learned that Matt's going to be moving on to Melbourne to an institute there to start his own lab. So best of luck in your future endeavours, Matt, and maybe we'll see you back in the UK sometime soon.
Excellent. Thank you very much.